please meet my colleague, Dr. Simon Burgener. Simon started his journey in biochemistry at ETH Zurich. Then he did his PhD at Max Planck Institute for Terrestrial Microbiology with Professor Tobias J. Arb on expanding the repertoire of enzymatic CC bond formation with 1 carbon units. After successfully defending his thesis, he has moved back to Switzerland to start his postdoctoral studies in the group of Tom here at the University of Basel. So Simon, the floor is all yours. Thank you.
Thank you very much, Manchista, for the kind introduction. I assume that you see my screen now and I'm set to go. Is that correct? All right. Yeah, thank you also to the organizers for giving me this great opportunity to present some of my research that I had been doing during my PhD in Toby Erb's lab. And the title of my talk is Expanding the 1-Carbon Chemistry of Thiamin-Dependent Enzymes. So first off, why should we be interested in 1-carbon chemistry? Well, I'm pretty sure that you all are familiar with this challenge that we are facing in the 21st century, that is increasing CO2 levels in the atmosphere and associated climate change. So on the one hand, we have this problematic molecule of CO2 that is accumulating in our atmosphere, causing global warming. But on the other hand, you could also view CO2 as a valuable carbon source to build up carbon molecules that are important to life on Earth. So in all these molecules, you find several carbon-carbon bonds, and you need a lot of carbon to build up these molecules. So if we could find efficient processes to turn the CO2 back into useful products, we could address this carbon challenge and find new ways to try to decrease the atmospheric CO2 levels and try to rebalance the global carbon cycle. And I, as a biochemist, I'm pretty sure that we can use enzymes for this task, because we looked into databases and we found that there's more than 300 unique enzymes that are involved in making or breaking CC bonds. So I think there's a great potential for enzymes to help us with this problem. But in a more basic conceptual way, we looked at CC bond formation reactions, because eventually when you want to fix CO2, you need a CC bond forming reaction, right? And if you look at it at a very basic level, you have an electrophilic carbon center reacting with a nucleophile, carbon nucleophile, in a carboligation reaction to make such a multi-carbon compound. So now if we want to use C1 molecules, which eventually derive from CO2, we can look at C1 electrophiles first, and we see that actually CO2 itself is, of course, is an electrophile, or also formaldehyde drawn here. And reactions of this type are quite easy, because these are readily electrophilic. It also turns out that there's a lot of enzymes found in nature that can do this trick. So almost all of these 300 enzymes that I talked about, so more than 200, can actually do that. So you have carboxylases or also decarboxylases that can run in reverse, lyases, aldolases, methyltransferases, and many more enzymes. So nature has really found many solutions to use C1 electrophiles. But on the other hand, if you look instead of using a C1 electrophile, use a C1 nucleophile, you find that this is quite challenging. And part of the reason is that none of the naturally occurring C1 molecules is very nucleophilic by nature. So perhaps you could say carbon monoxide would be somewhat nucleophilic on the carbon, but even that is not very reactive. So there are some enzymes, but it's actually roughly 10 enzymes in total that we know of that can use such a C1 nucleophile. And these enzymes typically require activation of the C1 molecule by means of organometallic species, which are then mostly oxygen sensitive, so not extremely practical in terms of biocatalytic use, or they also use expensive cofactors. So we were wondering if we could actually fill in this gap, so sort of trying to find a balance or try to find new enzymes that can use C1 nucleophiles, thereby potentially creating novel biocatalytic routes or even eventually synthetic pathways for carbon fixation. And we became interested in this enzyme family. So we looked into a lot of different enzyme families, but we found this superfamily of oxylyl-CoA decarboxylase and 2-hydroxy-acyl-CoA lyase, which split into these two phylogenetic clades. And what these enzymes have in common is that they use thiamine pyrophosphate as a cofactor. And TPP is one of nature's most abundant catalyst or cofactor for CC bond breaking or also making. So how the enzyme uses this cofactor is that in the active site, it gets deprotonated, forming this ylid structure, which is in resonance with a free carbene within the active site of the enzyme. This carbene can then attack onto a electrophilic carbonyl, as such as is the case in these enzymes, so either oxalyl-CoA or 2-hydroxyacyl-CoA. After decarboxylation or a cleavage of the CC bond in this part here, you form this intermediate, which is also referred to as a Breslow intermediate. And what is the most striking feature of this intermediate is that the carbon that was formerly electrophilic is now reversed in polarity to be a nucleophile. And these enzymes then protonate this intermediate and release eventually formic-CoA as a product. So you could argue that in a way this intermediate here is a one carbon nucleophile because you can use formic-CoA as a donor. So we raised the question: could this enzyme or could we use these enzymes to run this reaction in reverse? So using formic-CoA as a sort of activated formic acid, which is a C1 molecule, could we use this in reverse to furnish a novel type of CC bond formation? So condensing it with an aldehyde to give us a new 2-hydroxyacyl-CoA ester. Before we could start out, we wanted to investigate a little bit more in detail these enzyme families, because when we started, only two representatives or one representative of each clade had been characterized thoroughly. That is the human HACl, which is down here, as well as a bacterial oxalyl-CoA decarboxylase up here. And we selected a few candidates from these different phylogenetic clades, and we found that these oxys actually produce very well in E. coli, so you can get a high amount of protein by normal expression conditions, and also crystal structures are available for these enzymes. On the other hand, you have these HACLs, which you can actually get only very little protein yield from standard conditions. So these are poorly expressed, and there is no crystal structures available. I mean, you can get out some protein if you tweak the expression conditions, but for biocatalytic use or use as an enzyme in a pathway, this was not very promising. So we decided to stick with one of these enzymes here, and specifically I chose the enzyme from Methylobacterium extorquens. So first off, we just wanted to see if this enzyme can actually do this carboligation reaction. And in initial screens, we had actually observed that benzaldehyde can indeed serve as an electrophile, so thereby forming this product here, mandelyl-CoA, which is a derivative of mandelic acid. But of course, this enzyme is also still, or what this enzyme has actually evolved for, is decarboxylation of oxalyl-CoA. So when you incubate the enzyme with oxalyl-CoA and benzaldehyde, you find that the enzyme instantly decarboxylates the oxalyl-CoA, so in orange, to formic-CoA, and then slowly converts this formic-CoA into the mandelyl-CoA by the carboligation reaction. So we were wondering if it would be possible to improve this reaction. So because of course this rate here, 0.05 per second, is not very fast, especially comparing to the roughly 100 per second turnover rate for the decarboxylation. So we were wondering if we could change the active site of this enzyme to promote the carboligation reaction over the release of formic-CoA. And we had a look at the active site, and you find that water molecule is actually the proton donor in this final step of the reaction. Of course, you cannot mutate a water molecule, but what we did is mutate the coordinating residues, that is this tyrosine 497 and serine 568. And when you mutate these to alanine, you see that the KCAT drastically drops down to roughly 1% of the wild type in the case of the YA mutation, while the KM for oxalyl-CoA stays roughly the same. I mean, there's a slight increase, but it's not significant that you would think that the oxalyl-CoA is binding way worse in this mutant. So now we slowed down essentially this protonation step. So the next question was, is this enzyme now also better at doing the carboligation? And it turns out that it is. So when you now use, again, oxalyl-CoA as a donor, you see that it only slowly gets converted into formal-CoA, this is the green line here, and only to a very slow amount, but the predominant product in this reaction is actually mandylyl-CoA. So by introducing this one mutation, we slowed down the decarboxylation and we improved the carboligation rate. So in total, what we did by this one mutation, we did a 400-fold switch in carboligation versus protonation rate. So this was quite cool for us to see that we can change the active site of this enzyme to go from a decarboxylation reaction into a carboligation reaction. But of course, so how could this be any useful? How could we make use of this process? Because of course, CoA esters are not very biocatalytically relevant substrates nor products, because you cannot buy these CoA esters, nor is there a market to sell these products. So I thought about closing the catalytic cycle here in a way that we regenerate the oxalyl-CoA as a substrate for carboligation, and at the same time cleave off mandelic acid. And mandelic acid in turn is a very important chiral building block in organic synthesis, but it can also be found in many cosmetics. So this is a very useful product, and finding a way to make this with an enzyme would actually be a cool alternative. Because so there are ways to make these with enzymes, but those enzymes actually are all relying on cyanide as CC1 nucleophile. And working with cyanide is, I mean, it's possible, but it's also not extremely safe prospect. So finding an alternative could be also a cool thing to do. So we set out to find enzymes that could close this cycle. And first, I found a thioesterase from E. coli, this YCIA enzyme, that can actually cleave the mandylyl-CoA into mandylate and CoA. And it does so quite specifically. So of course, we have two other CoA esters in this cycle, so oxalyl-CoA and potentially formal-CoA, but those are not much affected by the thioesterase. So it's rather specific for mandylyl-CoA. Then I also found a CoA ligase that can take oxalate and ATP and turn it into oxalyl-CoA and AMP, and it's also a very efficient enzyme. So with all these enzymes in hand, I went on and combined them in a one-pot cascade. So I took all three enzymes, added the substrates, oxalate, ATP, and benzaldehyde, and just detected mandylate production over time. And as you can see here, the mandelic acid is forming over time in this catalytic process. And what is most striking here is that when you compare the wild type to the mutation I had introduced before, you see that the mutation causes a 5-fold increase in the rate of the reaction, and also a 4-fold conversion, higher conversion at the end of the reaction. And in numbers, so if you calculate the turnover number based on the oxy concentration, this is a 1,100 turnovers, and it does at the rate of 15 per minute in the first 3 hours. And this this mutation probably causes less formal-CoA to be released. As I have shown you before, the enzyme does not release much formal-CoA when it has this mutation, and the formal-CoA production is actually somewhat problematic because formal-CoA can hydrolyze spontaneously to some extent in water, but also can be cleaved by the thioesterase, thereby generating a futile cycle where you just burn through the ATP without making any mandylate product. So the mutation was really important in creating this catalytic cycle. What I had not talked about so far, but you might have noticed, is that the carboligation reaction actually creates a stereogenic center at the C alpha carbon here. And so far we had not known what would be the outcome of this reaction, if there would be any stereoselectivity. And doing chiral HPLC, we found out that the enzyme is actually predominantly producing S-mandelic acid with an enzymatic excess of 97% in the case of the mutant. It is important to say that the selectivity is actually determined by the ligase, because you could think that the thioesterase could also have an enantiomer specificity, but as I've shown you before in this plot here, I used racemic mandylyl-CoA here, and it's cleaved completely, indicating that both enantiomers are cleaved by the thioesterase. So as a consequence, the selectivity is really determined by oxy alone. Okay, so with these promising results in hand, we thought maybe we can take it a step further and also try other aromatic aldehydes as substrates. So we screened a whole panel of of aldehydes, including heteroaromatic aldehydes. And we see product formations in most of these cases. And for some of these, so the four on the top right here, I've investigated in more detail. And we see again this mandelic acid with 99% EE under these conditions with 75% conversion. But for example, also this chlorinated substrate with very high EE and also moderate conversion. Okay, so that's the first part of my talk, and you could now raise the question. So now I use oxalate and benzaldehyde, so both carbon molecules with more than one carbon. So how is this going to help us address the CO2 challenge? And of course, it's not really going to be helpful in that regard. So we thought in the next step, could we use formal-CoA and instead of oxalic-CoA as a C1 donor, and condense it with formaldehyde, which can also be derived from CO2 by reduction. And this reaction would then give us a C2 molecule that is glycolyl-CoA. We termed this reaction a glycolyl-CoA synthase reaction because the product is glycolyl-CoA. We again found initially that the enzyme is indeed able to do this reaction. However, the rate is very low, so you see a kcat of 0.2 per second and an extremely high Km for formaldehyde of 100 millimolar. And if you think about formaldehyde as being very toxic to most cells, a Km of 100 millimolar means that you need such high levels of formaldehyde that this would probably never become feasible in a physiological setting. So we thought maybe we could improve the enzyme by engineering in order to increase not only the affinity for formaldehyde, but maybe also the turnover rate. And for this, we turned in this case to a directed evolution. So we found in the active site of this enzyme 9 residues that shape the substrate binding pocket, and the 8 residues on top here are very close to what would be tentatively the place where the chemistry would happen of condensation of this intermediate with formaldehyde. So we targeted these 8 residues for mutagenesis, and we decided to go for an iterative saturation mutagenesis strategy. For this, we established a high throughput screen that is based on coupling several enzymes. So we have first our OXY enzyme of interest, which makes glycolyl-CoA. The glycolyl-CoA is cleaved to glycolate, and this CoA transferase ABFT at the same time regenerates formal-CoA from formate. So you also regenerate the substrate for the very reaction. The glycolate is then oxidized to glyoxylate with this glycolate oxidase, which at the same time produces hydrogen peroxide. This can then be used by horseradish peroxidase to create a fluorescent readout for this reaction. So in overall, we basically couple the oxy reaction to resolution production, which you can detect in a plate reader. So we screened then all these libraries. So in iterative saturation mutagenesis in the first round, we created all libraries at all 8 sites containing all 20 amino acids. And what you can see in these plots is on the x-axis the maximum slope of such a reaction, and on the y-axis the endpoint. And simply speaking, what this means is that on the upper right corner, these hits should correspond to a variance with increased activity. You can also see in white squares the wild type, so the parent enzyme, always in the lower left corner of these plots. So we ran through several rounds of iterative saturation mutagenesis. And at the same time, we kept lowering the formaldehyde concentration. So you can see in the first round, we used 50 millimolar of formaldehyde. And in the third round, we went down to 25 millimolar. And eventually, we went down to 10 millimolar. So the rationale here was that we thought that by using subsaturating concentrations of formaldehyde, we might be able to find variants with improved Km for formaldehyde. So after 4 rounds, we could not obtain any further improvements. So you can see in round 5 and in round 6, the wild type is actually, or the parent enzyme is actually the best performing in the screen. So after 4 rounds, we ended up with this quadruple mutant that we termed MEOXY4. And of course, so far, we had not characterized these in detail. So next we did a detailed characterization of the final version, but also all the intermediate variants. And we found that these, all the variants actually still expressed in very high levels, which was important to check because in our screen, we did actually not account for protein expression levels. So it could have been that the improvements that we had seen in the screen came about mostly by improving the expression, but this seems not to be the case, as all these are similar in expression levels. So next was to measure kinetics for these enzymes, and we found actually a 200-fold improved catalytic efficiency for the glycolyl-CoA synthase reaction. So that is the reaction that we had screened for. So from roughly 1 per molar per second, we went up to almost 400 per molar per second for glycolyl-CoA synthase reaction. At the same time, the enzyme sort of forgot how to catalyze its native reaction, that is oxalyl-CoA decarboxylation, because in the final variant, MEOXY4, only retained 0.1% oxy activity. So there seems to be a trade-off in these two reactivities in this enzyme. And in numbers, this means a 100,000-fold switch between these two reactivities. So next, we were also wondering if the enzyme can also use other acceptor substrates. So not only formaldehyde, but also acetaldehyde. And for example, also phenylacetaldehyde, which you can see here, were actually used as substrate. And the MEOXY4 showed increased activity with all the tested substrates. And what is also remarkable is, for example, MEOXY4 now also became a sort of efficient catalyst for using acetone as an electrophile, whereas the wild type had hardly any activity. So of course, this is a log scale in the K-ops here, so meaning that this is a really high improvement for this enzyme, which came about through directed evolution. So we also solved the crystal structure of the wild type and the mutant. And you can see here that you have the tyrosine to phenylalanine mutation that opens up the active site here to some extent. But the most striking mutation is arguably this glutamate to glycine, which opens up sort of a cavity around the TPP cofactor. And we think that this might cause this adjacent tyrosine to move over into this cavity, or give it some wiggle room to go there, and potentially then being involved into proton transfer reactions, which are important throughout this reaction. So next, we wondered if we could use this enzyme in a more physiological scenario, because eventually, if we want to use it as a carbon fixation catalyst, it would be really good to have a cell fixing the carbon, not using this in vitro with purified protein. So we teamed up with the group of Ramon Gonzalez at the University of South Florida, because they had established a system where they use a whole cell catalyst. So they have an E. coli strain that is engineered for the conversion of formaldehyde into glycolate. So their system also relies on an HACl enzyme, which takes formaldehyde and formal-CoA, makes glycolyl-CoA out of it, and the glycolyl-CoA is converted inside the cell to glycolate as the final product of this sort of mini cascade. And in their system, as we had seen before, the enzyme they had used so far, which was the best performing they found, was this RU-HACl, and you can see hardly any expression, whereas these OXY variants are highly expressed in their system as well. So this reflects the observations that we had made. So next, they also tested this productivity of glycolate in the strain and compared it to their best performing variant. And you can find that the in vivo results, or this productivity, is actually nicely reflecting the trends that we had seen measuring steady state kinetics with purified enzymes. So you see here increasing over the rounds is the catalytic efficiency, but at the same time also the productivity in vivo. And this was really promising for us to see that by using directed evolution in an in vitro setting, you can actually get higher productivity in a in vivo system. And this brings me to the end of my talk, just quickly summarizing. So I hope I could show you that by using enzyme engineering and rational design or directed evolution, it is possible to turn an enzyme such as a decarboxylase into a promising C1 carboligase. And then especially also combining this catalyst with other enzymes, we can create cascades, thereby giving rise to novel biocatalysis and metabolic pathways. And I'm sure that in the future we will be able to find more such enzymes that could help us with the CO2 challenge. Finally, I would like to thank some people. So the whole herb lab for creating a really nice atmosphere to work in. Most importantly, of course, Toby Herb, who you can see in this picture here. So this is, of course, a pre-COVID lab picture. I also would like to thank the collaborators at the University of South Florida, as well as the funding agencies. And of course, most of all, I would like to thank you for your attention. Thanks.
Okay, thanks very much, Simon. It was a fantastic talk. I'll pass over to Anya for the Q&A.
Yeah, thank you so much, Simon. It's great to see all these mini cascades, and I can only imagine, you know, like the subsequent reactions that you can start weaving into them. So I was just curious if you could comment a little bit on your ATP recycling system and whether you found some new ways of recycling ATP. And my second question was about the CoA, whether you could replace it with something simpler than a thio-CoA.
Yeah, so first the ATP, I have actually not used recycling in these screens here and all the data that I have shown you, but I actually did also a sort of semi-preparative scale of this reaction where I used higher concentrations and just a bigger volume. And there I actually used ATP recycling system, because in such a system, when you scale up, you have to use so much ATP that it's just not very economical anymore. And in those cases, I have used creatine kinase and creatine phosphate as a phosphate donor to replenish the ATP. For CoA, so yeah, CoA is also extremely expensive. So if you use it in a in vitro setting and you just add it to the reaction, this actually costs even more than using the ATP, even though the CoA can be used in catalytic amounts. So this is a major problem. And that's also why we actually now try to put this whole thing also in a whole cell catalyst, because a whole cell catalyst could not only deliver us an ATP pool. For example, in E. coli, you could just use glucose as a sort of ATP source, but it could also provide us with a CoA pool. So then we wouldn't have to use super expensive CoA. So this would be our way to tackle that problem.
Yeah, thank you. I see that Brandon has his hand up. So Brandon, do you want to unmute yourself and ask a question?
Yeah, sure. Thanks. Awesome talk, Simon. I love these enzymes. They're like an absolute gold mine. So I was curious to know if other types of acceptor substrates, maybe like oximes, were compatible with these types of enzymes. Just think it would be super cool.
Yeah, you mean in terms of electrophile, right? Yeah, so I have actually, I tried, what was it? Nitrobenzene, so like a nitro group, which can potentially serve as an electrophile. And I have seen some product, but I did not really go further into it because the analytics were a bit tricky. But I think the product will be a carbamate, if I remember correctly. So this would be potentially be something interesting, but I did not follow up on it. Another possibility that I didn't check is actually, I'm not sure if you could try to use alpha beta unsaturated ketone or aldehyde. That would be like sort of a Stetter type reaction, but I did not try that either.
Great, thanks.
Thank you.
Anya, I could go if that's okay. Simon, just obviously some of the like, so for example, BAL is able to dimerize aldehydes, right?
Yes.
What are the prospects of doing artificial formose reactions where you dimerize or trimerize formaldehyde directly?
Yeah.
In a controlled fashion, possible, or it's just not going to happen?
It is. I mean, it's a great question. It has been done. I think actually more than 5 years ago, the labs of, I think it was, I don't remember, but it was Justin Siegel was the first author on this publication in PNAS, where they used computational enzyme design. I was in collaboration with David Baker, where they actually turned benzaldehyde lyase, so BAL, into a formulase. So it catalyzes the condensation of 3 molecules of formaldehyde into dihydroxyacetone.
Mhm.
It's of course a super interesting enzyme, and would be a very powerful catalyst to turn C1 molecules into C3 molecules. But even though they had done design and several rounds of directed evolution, their final catalytic efficiency was something like 2 per molar per second. So very slow enzyme.
Mhm.
And yeah.
Yeah, you have probably really high KMs for the aldehyde. But yeah,
that's most of the problem.
But as someone who's worked in a sort of a C1 specialist lab, is that pathway an attractive route to, to getting C1 chemicals into carbohydrates and things? Is that an energetically the best pathway, would you say?
Which one? The formulase or formose? Yeah, it is actually. It should be. I mean, it's thermodynamically favorable, I think, the whole condensation reaction. And dihydroxyacetone is actually after a phosphorylation to dihydroxyphosphate. Dihydroxyacetone phosphate is actually central intermediate, so it's actually part of glycolysis. So you can enter central carbon metabolism directly. So it would be actually pretty interesting reaction, yes. And people have worked on this, have followed up on it, and there's many papers actually recently came out that have used the formulase enzyme, even though it's so such a poor catalyst really at the moment.